Good afternoon. Welcome to Dr. B.R. Ambedkar Open University. This is course 7, Toxicology of Insecticides. In this, we have 16 units. In the earlier classes, we have dealt till 14 units. And today, and this is the last class, we'll be dealing with 15 and 16th unit. So with that, you will be finishing your syllabus. And coming to the unit 15, it is insecticide resistance, and 16 is pesticide residues. So let's dive into the class now. I'll share the document. Yeah, so this is uh, insecticide resistance, the unit 15. In this chapter, we'll see the status of insect resistance to insecticides, particularly, and various mechanisms that are involved in the development of insecticidal resistance, its genetic basis, and the factors that are conducive to the rapid development of insecticide resistance, particularly in insects. So in this graph, you can see the chronological increase in number of cases of resistant species. So basically, what is resistance? So resistance to insecticide can be called as the development of an ability in a strain of an insect to tolerate doses of toxicants, so which would be lethal to it. So it will develop an ability to tolerate the lethality of the insecticide. So it will no longer be susceptible to the insecticide, but it will start accepting it and it will start bearing it. So it becomes resistant to that particular insecticide. So developing that ability itself is called as resistance. And resistance to insecticide was first documented in 1914 by A.L. Melendo in the scale insects to lime sulfur. So they are resistant to lime sulfur. These scale insects are found resistant to lime sulfur. Between 1916 and 1946, 10 more cases of resistance, particularly to inorganic pesticides like arsenical, selenium, cyanide, and other compounds were recorded. Then, so these are regarding the inorganic pesticides. Coming to the organic pesticides, also there is data reported around 1947, and that is particularly for DDT. And the resistance was developed by the houseflies to DDT was documented in 1947. So resistance is most acute in insects and mites, among which at least 447 species, including most major pests, have been reported to be resistant to one or more classes of chemicals. So it is not like this insect will be specifically resistant to only this kind of insecticide, but there are chances like mm, different species can be resistant to multiple insecticides or most classes of the chemicals. So in this, you can see the chronological increase in the number of cases with the years. So in the 1940, so the resistance is very less. Even the use of the insecticides was less. Uh, but gradually, as the years progresses, so you can see an increased resistance upon uses of increased insecticides towards the insects, crop, or whatever the purpose of it. But if the usage is more, so definitely the insect will get adopted to it and it develops an ability called resistance. So development of insecticide resistant traits. So how does this develop? So it should be well understood that the insect does not induce any heritable changes, but it merely selects out the resistance once over generations. So in the initial generation, you don't get it, but once over the generation, you tend to be resistant and provide an opportunity to the already existing innate sensitivity found only in a few individuals. So this means that in a given population, there are always individuals that can withstand certain amount of dose, which a large number is unable to do so. So in the earlier classes, when we have discussed, we have studied the bell curve. You remember that, right? In the normal distribution. So on the tail parts, at the left and right sides, so you see the most susceptible group which are extremely sensitive to the doses, and also you can see the most resistant group means they can even tolerate the higher dosages, and majority of them fall in the middle one, middle, where the 95% of the people or 99.5% of the people there are, they react similar to the uh, particular doses, where you can fix the particular dose like lethal dose 50 or something like that, right? So in this case also we are trying to explain the same concept, where this means that in a given population there are always individuals which they can withstand certain amount of dose, which a large number is unable to do so, so they are resistant groups, and development of such resistance and resistant populations is a case of simple Darwinian selection. Okay, so it is not well understood as to how few individuals in a population have the ability to withstand the insecticide, while a large number of are unable to do the same. So very few are able to withstand the toxicity or higher concentrations of insecticides, while larger group are not able to do so. Right. So, if you take the example of a COVID, like majority of them have fallen sick for the COVID, right? So, majority of them have got COVID, but there are very few people who did not even get affected, who did not even have a cold. So, they tend to be like more resistant towards that particular virus. So, in a similar way, the resistance in the insecticides is also seen for the insecticides is also seen, and this presumed to occur due to, so how does this happen, why few members are very much resistant towards the toxicity while majority of the people are suffering with it. So there are like two uh, presumed to occur due to natural mutations or gene amplification. So these two can be the reason. Of course, there are multiple factors involved, yet these two have been well studied, and these like a Darwinian selection, as we have said. So it's like a natural mutation or gene amplification. So what is a natural mutation? So insecticide resistant genes have an altered function. Example, altered juvenile hormone receptor that produces methoprin resistance in fruit flies, for example, or the alteration of glutathione transferase to catalyze glutathione dependent dehydrochlorination of DDT in house flies. So in fruit flies, you see JH receptors, and in house flies, you see DDT, glutathione dependent dehydrochlorination. So a particular species has resistance towards particular compounds, and it is dependent on the natural mutations, and it's a spontaneous genetic changes that can result in resistance to insecticides, and these mutations can alter the target sites of insecticides or enhance the detoxification pathways, and such examples are mutations in target genes like acetylcholinesterase, leading to reduced binding affinity for organophosphates, and examples are the mysis. Yeah. In the mysis periscae, you can see the green pitch aphid showing resistance to organophosphates and pyrethroids due to mutations in target proteins. And the other mechanism is gene amplification or hypomorphic mutation. So this is an insecticide resistant strains can also be caused by another type of gain of function mutation known as gene amplification. So it is not a loss of function, but it's a gain of function. So gene will be amplified. So it becomes more. So that's a kind of gain of functional mutation can be seen in this case, which refers to the genetic duplication of a normal gene. So that is a gain of a function mutations. And multiple copies of a gene, when activated, result in an increased synthesis of the corresponding mRNA, further resulting in an increased synthesis of the corresponding protein and a net increase in the total enzymatic activity of the protein. So obviously, if the mRNA increases, the protein synthesis increases, and the total enzymatic activity of the protein will also be increased. And this process where multiple copies of a gene are produced, leading to overexpression of the encoded enzyme or protein, which can confer resistance, is observed in gene amplification. So the overexpression of, for example, the overexpression of detoxification enzymes such as CYP450s that leads to increased breakdown of insecticides is an example of gene amplification where overexpression can be seen. An example organism is again the same mysis has developed resistance to pyrethroids through gene amplification of P450 genes, which enhances the insecticide detoxification. So the mechanism of initial gene duplication mutation is not well understood, although such events are thought to have occurred frequently in evolutionary history. Also, gene amplification can be reproducible, induced by drug selection. And genetic duplication events probably occur more frequently than neomorphic mutations. And the other reasons can also be so, other than the natural mutations and gene amplifications, a target site mutation can also develop insecticide resistance, like genetic changes in the insect's target site that reduce the efficacy of insecticides, making them less effective. Example, mutation in the sodium channel gene. What will happen if there is a mutation in the sodium channel gene? It will reduce the sensitivity of the target to pyrethroids, thus causing resistance. Again, the example is Mysis persicus has a mutation in the voltage-gated sodium channel, conferring resistance to pyrethroids, which act on that particular channels. An overproduction of detoxifying enzymes can also develop insecticide resistance. So what does overproduction of detoxifying enzyme means is enhanced production of enzymes that degrade or neutralize insecticides such as CYP450s, glutathione S transferases, or carboxylesterases, like that. And examples, they increase the levels of P450 enzymes that break down insecticides like pyrethroids and neonicotinoids. And again, mysis has developed resistance to neonicotinoids through the overproduction of 450 P450 enzymes. And cross resistance can also be the reason for development of insecticide resistance. And resistance to one insecticide confers resistance to others. That is cross resistance. So if an insect is resistant to one insecticide, it can also confer to the resistance to the others, often due to their similar biochemical pathways that they share, or the same similar detoxifying enzymes that are involved in both the insecticides or the other insecticides. So this also will confer the resistance to not only one insecticide, but to also to the other insecticides which share the same biochemical pathways and detoxifying enzymes. So that is cross resistance. So resistance to one class of insecticides like pyrethroids, leading to resistance to another class like neonicotinoids, because they have similar detoxification mechanism. And again, we have commonly taken the example of mysis for better understanding, and it shows cross resistance to multiple insecticide classes like pyrethroids, neonicotinoids due to the P450 overexpression. Coming to the mechanism of insecticide resistance, so basically what resistance is the reduction in the sensitivity of an insect population, right? So they become resistant. I mean, they don't respond, or they develop an ability to not get affected by that. So that is resistance. So that is reflected in repeated failure of a product to achieve the expected level of control when used according to the label requirement. So if we will say like pyrethroid, if it is given in for an example, you say like 1 mg per gram body weight in normal mammals, then it will be toxic, or it causes lethal dose 50, or it is like very much toxic. But that is not happening, because that 1 mg is not sufficient to cause lethal dose 50, because that particular mammal, they have developed resistance to pyrethroids, which does not act efficiently on that particular organisms. Right. So, however, the problems of product storage application and unusual climatic or environmental conditions must be also eliminated when you're dealing with the insecticide. So this can also cause to the less efficiency as well. So there are several ways insects can be resistant to crop protection products, and pests often utilize more than one of these mechanisms at the same time. So you can't predict like only due to their behavioral resistance, or only due to their cuticular penetration resistance, or only due to their metabolic resistance, like this insect got resistance towards that particular insecticide. But there are multiple mechanisms that are involved here, not only one. and what are they and how does it cause, that we will see with this. So here you have seen a caterpillar which is on the leaf, and how does this insect, how does the mechanism of insecticide resistance works here, you can see. So there are like major mechanisms of resistance, like behavioral resistance, penetration resistance, penetration through the cuticle, penetration resistance, knockdown resistance, metabolic resistance, or altered side of action resistance, or increased rate of excretion. So these all can lead to the insecticidal resistance. So these are all the mechanisms that leads to the insecticide resistance. Coming to the behavioral resistance here, you can see. So behavioral resistance is the inheritance of a behavior pattern that somehow causes insect to avoid a toxic dose. So insect will not completely encounter the toxic dose, but it will try to avoid the toxic dose. So resistant insects may detect or recognize a danger and avoid the toxin. So it is called as pseudo resistance. So when they sense the danger, they escape it and they avoid that particular path. So, in that case, also, it will not encounter the toxic dose. And this mechanism of resistance has been reported for several classes of insecticides, including organochlorines, organophosphates, carbamates, and pyrethroids. So, insects may simply quit feeding if they come across certain insecticides or leave the area where an insecticidal has been deposited. For example, insects may move to underside of the sprayed leaf, move deeper in crop canopy, or fly away from the target area, mm, so that it will avoid a toxic dose. And with transgenic plants, insects may stop consuming or reduce ingestion of enough toxin that can kill them. So that also leads to the survival and being resistance, that is behavioral resistance. When coming to the cuticle penetration resistance or pen resistance, so penetration resistance occurs when insects can slow absorption of the chemicals into their bodies because their outer cuticle has developed barriers against that particular insecticides. So penetration usually is not a major factor in resistance and is present along with other forms of resistance and intensifies the effects of those other mechanisms. So it can be more readily measured by topical application of labeled insecticides to both susceptible and resistant individuals. And then measuring the amount of remaining insecticides on the cuticle after a specified period of time will also be seen in this. So, a slow penetration only retards death in the absence of mechanism like detoxification. So, it is dependent on the interaction with the detoxification if there is to be an effect on the lethal action of the insecticide. A difference in penetration has also been reported as one of the R mechanisms in dimethoate resistant Panonicus citri and pyrethroid resistant cattle tick and other cases as well. So, decreased cuticular penetration of pyrethroids is considered to be a mechanism of resistance, particularly in houseflies. And knockdown resistance, if you'll see the knockdown resistance, so insects with knockdown type of resistance, they do not respond to a dose that normally kills the susceptible strain, or symptoms of poisoning take far longer periods than this kind of insects. So most important resistant mechanism for those pyrethroids wherein toxicity remains unaffected by synergesis is the knockdown resistance. So it is mostly seen in pyrethroids. So which results in a broad cross resistance to pyrethroids, DDT, and its analogs. So one or more recessive genes present on chromosome 3 governs this, and this mechanism is not due to increased metabolic degradation or decreased penetration, but it is due to the nerve insensitivity. And coming to the metabolic resistance, uh, here you can see that in metabolic resistance, resistant insects detoxify or destroy the toxin faster than the susceptible insects, or quickly rid their bodies of the toxic molecules. So, metabolic resistance is by far the most common and complex mechanism and often presents the greatest challenge. Insects use their internal enzyme systems to break down the insecticides. So that is a major challenge here. And resistant strains may possess greater levels or more efficient forms of these enzymes. In addition to being more efficient, these enzyme systems also may be broad spectrum, meaning they can degrade many different pesticides. The various mechanisms can be present alone or in combination, and the easiest adaptable species can finally build up to complex multiple resistant strains that can resist the majority of the insecticides. Nature has provided the insects with enzymes that alter, digest, or break apart organic compounds. So, even before insecticides were introduced, insects had the metabolic mechanisms to protect themselves against poisoning. And the classic example of this is the immunity of tobacco budworm towards nicotine present in the tobacco leaves. So, the analysis of the contribution of various detoxification mechanisms can be quite complex. In particular, since sometimes different mechanisms can produce the same degradation products. And coming to hydrolases, three important groups of insecticides, the organophosphates, phosphates, carbamates, and pyrethroids are esters and would seem susceptible to development of resistance by increased activity of hydrolytic enzymes. So far, this has only been shown to be of great importance for organophosphate compounds, whereas esterases play a smaller role in resistance to pyrethroids and practically none in that of carbamates. In the housefly, several strains resistant to parathion, diazinon, and a number of other organophosphate compounds have hydrolytic enzymes which act as phosphatases on the fever analogs such as paroxone. Coming to mixed function oxidases, so these are also involved in this. So the increase in the mixed function oxidase activity is one of the most common mechanisms of resistance to a great variety of insecticides. Even though compounds such as DDT and DDE can be degraded by certain R strains of the insects, specific inhibitors such as piperonyl butoxide and Sesamax, they facilitate the in vivo degradation, although they can sometimes be degraded themselves. So it should be borne in mind that a mixed function oxidases activity is dependent on complex system, the activity of which is determined by a reductase, one or more cytochrome P450s, and the concentration of the NADPH. So there is, in general, no proportionality between the rate of the oxidative degradation causing resistance and the overall amount of CYP450s or of one of its components as well. And in the see target site insensitivity, we can also see the altered side of action, particularly acetylcholinesterase. So the best example of an altered target as a cause of resistance to insecticides is that acetylcholinesterase, where we have been continuously speaking in all the pesticides, majorly with the reduced sensitivity to organophosphates and carbamates. So this ACHEs, that is acetylcholinesterase enzyme of this type, have been found in a large number of strains of arthropods, and the existence of a relation between insensitivity of acetylcholinesterase and resistance establishes that inhibition of acetylcholinesterase is indeed the mode of action of the esterase inhibitors. So in several cases, insensitive acetylcholinesterase in our strains have a lower activity than the normal ones, resistant strains. So in the other insects with altered acetylcholinesterase, there are no marked differences in enzyme activity. The reduced acetylcholinesterase activity could be due to reduced amounts of enzymes or to reduced catalytic activity. In a single gene is responsible for the difference in the acetylcholinesterase sensitivity and the resistance caused by it. In spider mites, cattle tick, green rice leaf hoppers, and houseflies. For the housefly, the gene has been shown to be located on chromosome 2. So indications of resistant acetylcholinesterase in several populations of houseflies and insensitivity of acetylcholinesterase are very much widespread. A tenfold increased activity of acetylcholinesterase has been reported in Drosophila strain selected with parathion. There is insufficient evidence that the enzyme hydrolyzing the acetylcholine is indeed an acetylcholinesterase. So, increased rate of excretion. In a classical experiment by Gotre et al., biosynthetic C14 nicotine was fed to tobacco hornworm, and in about 4 hours, 90% of the oral dose was excreted, and excretion was total in 1 day. So, when nicotine was injected into the body, 83% was recovered from fecus in about 15 minutes. So, the sensitivity of the tobacco hornworm to nicotine is attributed to its rapid rate of excretion. So, there is increased rate of excretion. And coming to the factors conducive to rapid development of resistance. So, what are the factors that induce a rapid development of resistance? Is the speed with which the resistance develops. So, what is the rapid development of resistance? That is the speed with which the resistance develops depends on several factors, including how quickly the insect reproduce, the migration and host range of the pest, the crop protection products, persistence and specificity, and the rate, timing, and number of applications they have made. So, these are all the factors that contributes, and resistance increases fastest in situations such as greenhouses where insects or mites reproduce quickly. There is little or no immigration of the susceptible individuals, and the grower may spray frequently. An example is the control of insects in Japanese rice fields, where the search for insecticides that are specific in their action against certain resistant strain of insect together with a change in cultivation methods has been observed. And important factors that are conducive to the rapid development of insects developing resistance to insecticides include a prolonged exposure to a single nuclear insecticide. So, if you keep on spraying the same insecticide, then definitely that particular insect will get resistance to that particular insecticide. So, there should be you should use you change the insecticide or use a mixture of insecticides. You have to do in such a way, but not a prolonged exposure to the single insecticide. And high selection pressure is another example, like insecticides with a high selection pressure leave out a small number of individuals that can withstand that molecule. An example of this is the use of synthetic pyrethroids, which are very potent and effective insecticides in some situations, eliminating over 90% of the susceptible population. A similar situation can be expected with a higher dose of an insecticide. So, management strategies resorting to higher doses of insecticide to combat resistance has this advantage. And another is the large coverage area, where insecticides applied over a very large area results in eliminating a very large number of susceptible individuals. So, lack of susceptible population reduces the possibility of diluting the resistant population. And another is the insecticides related to the one used earlier. Means insecticides belonging to the same group or those having the similar mode of action have the same effect as those after using the same insecticide continuously. So treatment triggered by low number of insect pests can also be the other reason, where treatment with insecticides when low number of insect pests are present, select resistant ones with low numbers, and in the absence of susceptible ones have a better chance of multiplication. And other things like selection occurring prior to mating, no gene flow between population, no migration, short life cycle of the pest, insects multiplying by asexual means, and numerous offsprings per season, and insects highly mobile or migratory can also be the reason. And selection at every stage of insect life cycle is also the other reason. Because in the if the same insecticide is used to control the pest at the immature and the adult stage, it is obvious that the selection is at every stage of life and development of resistant strains is faster. So malathion is effective against the larvae as well as the adults of the mosquito. So in areas where it was used in stagnant water for the control of larvae and also in houses for the control of adults. So resistance to malathion developed faster than in areas where it was restricted to one stage of the insects. So if you use the same pesticide in the larval stage and the adult stage, so definitely that insect will be resistant to that particular insecticide. And coming to the genetic basis of the resistance, so resistant mechanisms are generally dependent on single genetic factors. The R factors have been generally accepted as single genes, but some examples have now emerged in which a series of gene duplications has led to the clusters. And the genetic base of resistance involves cross resistance, multiple resistance, and induction. In the cross resistance, so one resistant gene selected by the treatment with a particular insecticide often also confers more or less resistance to a number of other insecticides. So that is cross resistance. Earlier we have seen in the same class. The cross resistance, if one particular insecticide is resistant to pyrethroids, it can also be resistant to neonicotinoids because they share the similar biochemical pathway and the similar detoxifying enzymes. So that is what happens with the cross resistance. And we have also seen the example of Myzus periscape. And coming to the multiple resistance, the term multiple resistance is used when a number of resistant mechanisms are present, often as a result of selection with different insecticides. So sometimes the various mechanisms each affect only their own selecting agent, but generally they contribute more or less cross resistance to the other compounds. So, since the different mechanisms often show interactions which significantly augment resistance, the term multiple resistance is sometimes used. So, multiple resistance is now present in many insects that have been under long lasting insecticide pressure. For example, in the housefly, spider mites, cattle tick, and Spodoptera species. So this can result in a continued rise of resistance to certain insecticides even after they have been replaced by others. So even though you change the insecticides, they tend to have that continued rise of resistance. So the explanation is that the new compounds further select for the same mechanisms that show cross resistance to the ones that have been abandoned. So that is the multiple resistance. Um, Coming to the target site resistance is also one here. So the resistance occurs due to genetic mutations in the target site of the insecticide, making it less effective or unable to bind to its target. Coming to induction. So induction is the increased production of enzymes caused by the presence of certain inducing substances derived from the food, treatment with chemicals, or normally occurring in the body. So it is an individual reaction to the substances, and the induced state is not inheritable. And metabolic resistance, so resistance arises from the enhanced ability to metabolize or break down insecticides via detoxifying enzymes. And genetic basis in this is gene amplification. Example, Helicobacter where it develops metabolic resistance to pyrethroids to increased POE450 activity. So how do you manage insecticide resistance? So you know you have so many reasons, so many other factors for conducive to rapid development of resistance, and also the mechanisms that are involved in the insecticidal resistance and genetic basis of resistance. So how to manage all of this and how to manage the insecticide resistance? So experience has shown that all effective insecticide and acaricide resistance management strategies seek to minimize the selection for resistance from any one type of insecticide or acaricide. So in practice, alternations, sequences, or rotations of compounds from groups with different modes of action provide a sustainable and effective approach to insecticide resistance management. So you have to use insecticides with different modes of actions, not having the same mode of action, because if you use the insecticides with a similar mode of action, what will happen? The cross resistance will develop. So that's the reason you have to use the insecticides with a different mode of action. Okay, this ensures the selection from compounds in the same mode of action group is minimized, and applications are often arranged into mode of action spray windows or blocks that are defined by the stage of crop development and the biology of the pests of concern. So you can also use you limit or minimal the use of the insecticides, or use alternate insecticides, or synergistic and co-toxicity of the insecticides can be reduced. And integrated pest management, that is IPM, can be employed to minimize the or to manage the insecticidal resistance. So in that case, the minimal use of insecticides is done like unlimited applications per year of the same insecticide against Spodoptera, not only on cotton but other crops as well, as had led to sequential development of resistance to most available insecticides in Egypt. So in Egypt, you have seen that, particularly in Spodoptera, upon continuous usage of the applications of the insecticide per year, it has developed the resistance not only on cotton but other crops as well, had to led to the sequential development of the resistance. So you should not use the same insecticide for a prolonged period of time, or in like you should not use it in a higher number as well. So the application should also be reduced, the dose should also be reduced. So minimal use of insecticides is always encouraged, so that this strategy provides to be extremely effective, and there has been no resistance in the field, although many laboratory experiments demonstrated a potential to develop strong resistance to pyrethroids in case of littoralis on cotton. As And during the 1983 to 84 cotton season, there was a good adherence to the strategy, and pyrethroid failures were very few will observed. And laboratory essays showed that about 10% of the individuals collected in the cotton growing areas were heterozygous for pyrethroid resistance, but homozygous were rare. Coming to alternate insecticides, you have to use alternate insecticides in where the widespread strong resistance to pyrethroids in houseflies of animal farms in UK resulted from a widespread sequential use of DTT and trichlorphon, since the former selected knockdown resistance and the later both mechanisms confer cross resistance to pyrethroids. So this can also happen in the similar way. So rotation can work provided there is no cross resistance in the process. So alternate insecticides should also be uh, given upon checking their mode of action, and whether they will be cross resistant or not. So that also has to be taken into consideration. And synergistic and co-toxicity of insecticides. So small amounts of pyrethroids increase the effectiveness of the organophosphate insecticides. So this will boost the effect of the other insecticides. As a result, commercial formulations containing mixtures of the two groups of insecticides have recently been introduced. And co-toxicity probably works through the inhibition of organophosphates and or pyrethroids of the same enzymes, presumably like esterases which hydrolyze both group of the chemicals. This indicates in such situations, mixtures may be preferable over rotation. A successful control of pyrethroid-resistant Colorado beetles was achieved with pyrethroids synergized with piperonyl butoxide. And that's one of the example. And coming to the integrated pest management. So rational pest control strategies must be designed to manage resistance, both to prolong the effectiveness of the pesticides and to reduce environmental contamination by excessive use of the chemicals. So the ultimate strategy to avoid insecticide resistance is its prevention. So that is the best you can do. So, so initially you try to prevent, then you avoid the insecticidal resistance. So in order to prevent it, you have to develop an insecticidal resistance, or can be delayed by the integrated pest management approach, that is IPM approach. An integrated approach to managing pests should be adopted, as many different control mechanisms as possible should be incorporated. So the IPM based programs include the use of synthetic insecticides, biological insecticides, and beneficial arthropods like predators and parasitoids for the use, and cultural practices, transgenic plants, crop rotations, and pest resistant crop varieties, and chemical attractants or deterrents are used. So IPM will merge all of these or use any of these in combinations in order to delay the resistance. So pest monitoring and economic threshold, time of application, application techniques, protecting protecting beneficial arthropods and preservation of susceptible genes are the part of the IPM management. So rational pest control strategies must be designed to manage resistance, both to prolong the effectiveness of the pesticides and to reduce environmental contamination by excessive use of chemicals. So these strategies should be based on IPM techniques. So here, high doses should be uh, minimized because they cause insect resistant management, which is shown in the figure. So with this, we are done with our 15th chapter. In this, we'll also see when genetically modified varieties are used. Ideally, this must be alternated with rows of genetic modified plants to allow multiplication of susceptible populations. And cultural practices like destroying crop residues can deprive insects of food, and this cultural practice will kill pesticide resistant pests and prevent them from producing resistant offspring. So these are all the general things that can be done along with the other strategies that we have discussed earlier. So from the 15th chapter or 15th unit, so what are the questions that you can expect from that unit is what are the different mechanisms involved in the development of insecticide resistance among insects, and list of various factors conducive for rapid development of insecticide resistance in insects, and what are the different resistant management strategies that can be adapted to delay the development of insecticide resistance in insects that we have talked like IPM and all. Okay, so with this, we are done with the 15th unit and coming to the 16th unit. This is going to be the last unit of your syllabus, 16th unit, that is pesticide residues. So with this, you will be done with your syllabus. So this is the last unit in your syllabus, pesticide residues. So in this, we will be knowing all pesticides are inherently toxic molecules and their widespread indiscriminate use of may leave the residues leading to human health hazards and environmental risks. So in this chapter, it is intended to provide basics of pesticide residues and the principles that are involved in the analysis of pesticide residues. So use spray pesticides to kill the pest. So is it finished there itself? Does the pesticide just kill the pest? That's it. The, does it not have any effect on the environment? Yes, the answer definitely is yes, because after killing the pest, it will be still on the plant or it will be still in the soil. It will get incorporated in the soil. It will not only be toxic to that particular pest, but it is also toxic to other beneficial microorganisms as well and its surroundings, and it will be accumulated in the soil or it will be in the penetrated into the soil or into the food that we consume if you spray it on the crops or on vegetables or whatever it is. So that residues, so whatever the remaining part it is there, so that is the residue, that pesticide residue, how does it act on the health of the environment and humans, and how do you analyze these pesticides is what we will see in unit 16, pesticide residues. So pesticide is a chemical that is used to kill pest organisms by virtue of its toxicity, and it includes all the specific forms of applications like insecticides, fungicides, herbicides, bactericides, and many more depending upon the species. So, however, the misuse and abusive or abuse of insecticides beyond recommendations has led to several unwanted problems. By their very nature, most pesticides inherently have risk of harm to humans, animals, and environment because they are designed to kill or otherwise adversely affect the living organisms. And pesticides effects can be both acute and chronic, which vary from pesticide to pesticide. So, when applied properly, the risk to people who are not involved directly in their use can be minimized though the risk of the applicator can still be there. So the doses received through casual exposure, such as by walking through a treated field, inhaling vapors from a treated area, or eating produce from the treated field, would rarely be sufficient to cause a health problem. So these pesticide residues, they cause acute and chronic. Acute in the sense, if it is in higher doses, then it will immediately show a toxic effect, or upon continuous exposure to this pesticide, you tend to develop an chronic resistance with that particular pesticide. So this is what the pesticide residues they do. So there are certain critical terms and definitions that we regularly use in toxicity of insecticides, and you have been gone through all the terms and definitions in one or the other chapters or units of your syllabus. But in this, they have given in the syllabus, so we'll just brush up the basics of what it is. Like, what is a pesticide? So pesticide is broadly understood as any product that kills or controls various types of pests that cause economic damage to human beings either directly or indirectly. Okay, that is pest. So what is the deposit? Deposit is the amount of initially laid down pesticide on the surface. So whatever the wherever it will go and stick to the surface, it means that it got deposited there, right? So the amount of initially laid on pesticide on the surface is called deposit. So what is pesticide residues? So now the chapter we are doing is pesticide residues, right? So what is a pesticide residue? So pesticide residue means any specified substance in food, agricultural commodities, or animal feed resulting from the use of a pesticide. So the term includes any derivative of a pesticide such as conversion products, metabolites, reaction products, and impurities considered to be toxicologically significant are known as pesticide residues. Degradation is a process by which a pesticide is broken down to simple structures through biological or abiotic mechanism. Synonyms include breakdown and decomposition. So degradation is also called breakdown and decomposition. Dissipation and persistence. So in nature, disappearance of residues takes place in two steps. The first step is the initial phase in which the disappearance of the residue is fast. This phase is called dissipation. The second phase, in which there is a slow decrease in the amount of residue, it is known as persistence. Dissipation means the residue will be disappeared faster. Persistence means the residue is still present and it's slowly getting disappeared. Half-life, it is the time in which half the amount of initial deposit is eliminated by reacting or otherwise getting dissipated. So half-life values or the 90 to 100% disappearance values are used to express the approximate rate of the residue disappearance. And coming to ADI, it is an acceptable daily intake. So ADI of a chemical is the daily intake, which during an entire lifetime appears to have an appreciable risk to the health of the consumer on the basis of all the known facts at the time of the evaluation of the chemical. Noel, it is like a non-observed adverse effect level. So it is the highest dose of the substance that does not cause any detectable toxic effects in an experimental animal studied. So it is expressed in mg per kg body weight per day. So this is the highest dose of a substance, but still you don't see any significant changes or any lethal changes or toxic changes. That is Noel. Tolerance level is the tolerance levels may be defined as the maximum level of pesticide residues that are legally permitted. It means you'll see, right, there are certain uh, ingredients in your food or the packaged food that you see, and they are like preservatives, colors, food additives. So they all have a certain uh, range. So if you use above the tolerated level, then definitely you will fall sick or it will be lethal to you. So there is a determined fixed amount or fixed concentration that has to be used. And coming to MRL is the maximum residue limits. So maximum residue limit may be defined as the maximum level of the pesticide residues present in or on a produce when pesticide is used under supervision following good agricultural practices. So GAP is good agricultural practices. So they include the nationally authorized safe use of pesticides under actual conditions necessary for effective and reliable pest control. LOD is the limit of detection, that is the lowest concentration of a pesticide residue in a defined matrix where positive identification can be achieved using a specified method. So this you see in the quantitative analysis. So you will see the limit of detection. LOD is the limit of determination. So limit of determination is the lowest concentration of the pesticide residue or contaminant that can be identified and quantitatively measured. Withholding period or waiting period or pre-harvest interval is the minimum permissible time between the last application of a pesticide to a crop and harvesting for human consumption or grazing with the livestock. So that is the waiting or withholding period. And coming to weight units, so in residue analysis, weights much smaller than the milligrams are usually used, and these are explained like micrograms, nanograms, or picograms. So micrograms is 10 power minus 6 grams, nanograms is 10 power minus 9 grams, picograms are even less, that's like 10 power minus 12 grams. There are certain terms like parts per million, parts per billion, and parts per trillion that have been used, and parts per million is the parts of a residue in million parts of a sample. Very commonly, you can say it like mg per kg is parts per million. And the parts per billion is the nanogram of a pesticide per gram of sample, or mg or micrograms per kg of sample is parts per billion. And PPT, that is parts per trillion, is 1 picogram of pesticide per gram of a sample, is 1 part per trillion, and 1 part in million parts. And coming to multi-residue method, so analytical method which measures the number of pesticide residues simultaneously is called multi-residue method. We will see certain terms like bioaccumulation, bioconcentration, and biomagnification. So bioaccumulation refers to the entering of pollutants into the food chain and process of their increase in concentration from the environment to the first organism in a food chain. Like how they will first start to accumulate is what we are seeing here. So initially they enter into the food chain slowly and process of their increase in concentration from the environment to the first organism in the food chain that they encounter. Bioaccumulation. So, biomagnification refers to the tendency of pollutants to concentrate as they move from one trophic level to the next, and process of increase in concentration of pollutant from one link in a food chain to another. So, these phenomena are of very much concern to the environmentalists because together they mean that even small concentrations of the chemicals in the environment can find their way into organisms and accumulate in doses high enough to cause problems, eventually affecting the environment drastically. Bioaccumulation is a general term for the accumulation of substances in a living organisms. For example, pesticides such as DDT or other organic persistent chemicals can accumulate in tissues, fat bodies, or any part of an organism. The accumulation process involves the biological sequestering of substances that enter the organism through respiration, food intake, epidermal contact with the substance, or other means. So basically, bioaccumulation is accumulation of a pesticide or a toxic substance in an organism over time from the environment. And key features is it occurs when an organism absorbs a substance faster than it can eliminate it. And the substance can come from air, water, or food that it consumes. And it occurs within a single organism throughout its life. Example, pesticide residues in fish that accumulate over time, leading to higher concentrations in the organism compared to the surrounding environment, like mercury levels. Example. And bioconcentration is the uptake of a pesticide from the surrounding environment, typically water, by an organism. Here also you can take the example of a fish in contaminated water accumulating pesticides such as DDT in their tissues due to direct contact with pesticide-laden water. And biomagnification, the increase in the pesticide concentrations as you move up the food chain from prey to predator, occurs as predators consume contaminated prey. The concentration of toxins increases with each trophic level. Toxins are stored in fatty tissues and can be passed along to predators. So the predator will be affected by the prey. So and it will be more in the predator compared to the prey itself. So birds of prey such as eagles accumulating high levels of DDT after feeding on contaminated fish or smaller animals. Coming to the effect of pesticide residues on humans and environment and non-target organisms. So health effects on human beings particularly. So the effect of the pesticides can either be acute or chronic. So acute in the sense it is an immediate response to short-term exposure to a high dose. Or chronic is a response to long-term exposure to a low dose. So examples of acute health effects including stinging eyes, rashes, blisters, blindness, nausea, dizziness, diarrhea, and death. So examples of known chronic effects are cancers, birth defects, reproductive harm, neurological and developmental toxicity, immunotoxicity, and disruption of the endocrine system. So in this you can see the pesticide residues, they affect the human health, wildlife, bird, fishes and other animals, beneficial insects, plants, water and soil as well. So pesticides vary greatly with respect to their mode of action, the range of organisms they affect, their acute effects, and their chronic effects. Some pesticides can pose a significant risk to human health or to the environment, while others present very little risk at all. So reading the pesticide label is the quickest way to obtain information regarding the relative risk posed by a pesticide. So coming to the acute health effects, depending on the product, pesticides range from low to very high in acute toxicity to humans. In general, among pesticides, insects as a group are more toxic to humans due to their metabolism difference and the enzyme difference and all. They are more toxic to insects than the humans. And chronic health effects include cancer and other tumors, brain and nervous system damage. And pesticides have been implicated in human studies of leukemia, lymphoma, and cancers of the brain, breast, prostate, testes, and ovaries. So you can see here the picture, the same thing is said. Health effects of certain classes of pesticides like organophosphates and carbamates. They attack the brain and nervous system, interfering with nerve signal transmission, which we have seen in our earlier classes. And organochlorines. So many banned pesticides, including DDT, are organochlorines and central nervous system stimulants that can cause tremors, hyperexcitability, and seizures. Stages of exposure. Um, Pesticides pose the greatest risk to the people who handle or apply them, the workers during manufacturing of the technical grades, as well as during formulations of the product, are being continuously exposed to the pesticides. Their blood cholinesterase levels are monitored by the industry for the exposure hazards. And an unintentional exposure through foods due to pesticide residues can also happen. So pesticide residues in foods and environment will pose a greater threat to humankind and as well as co-survivors. So this issue is now gaining momentum, and recent examples of Kerala news reports on pesticide residues in water, vegetables, and beverages led to formation of joint parliamentary commission to look into settling the tolerances. And it also has effect on the non-target organisms in two ways, by direct contact, or the pesticide may leave a residue that causes injuries later, and it will be harmful when it comes into the contact. Coming to the pesticide residue standards in food and water, maximum residue limits are based on recognized safe and reasonable pesticides use pattern. In USA, tolerance levels are used in place of maximum residue limits. And increasing attention is also being given to environmental issues, particularly to sensitive resources like water, air, and soil. So these are like basic things and very sensitive resources. So you have to make sure that they are pesticide free and there should not be any contamination. So all over the world, the increasing number of consumers and most governments are becoming aware of food quality nowadays. And it is now common for consumers to demand that their governments take legislative actions to ensure that only safe food of acceptable quality is sold in the market and that the risk of foodborne health hazards is minimized. So the first steps were taken in 1961 to establish a Codex Alimentarius Commission. The body charged with developing a food code to study the pesticides and their residues. Food and Agriculture Organization and World Health Organization have jointly formed a group called Joint Meeting on Pesticide Residues, which we call as JMPR. It assesses pesticide residues in food and provides recommendations for maximum residue limits for pesticides in food and feed. So this group is also responsible for reviewing toxicological related data for estimating the acceptable daily intakes. And this joint meeting on pesticide residues, JMPR, serves as a scientific advisory body to Codex Alimentarius through Codex Committee on Pesticide Residues. The severe regulatory process in recent years after World Trade Organization era led to serious discussion on pesticide residues in foods and their impact on health and environment. While Central Insecticide Board under Insecticide Act 1968 of Department of Agriculture and Cooperation, Government of India, has got the authority to notify residue tolerance levels. The enforcement of residue tolerance levels is by pesticide formulations and analysis as per Part 14 of Pesticide Formulations and Analysis. In India, prevention of food adulteration. So it is based on the Prevention of Food Adulteration Act. PFA, that's Prevention of Food Adulteration Act 1957, and it's the not pesticide formulations and analysis, it's prevention of food adulteration here. So you have to do this correction here. So any pesticides that might leave residues in a food is regulated by the Prevention of Food Adulteration to determine the safe amounts of pesticides that can remain on raw agricultural produce used for food. As per the Prevention of Food Adulterations, the maximum residue limits are set for 121 pesticides on various crops, and for 10 pesticides, maximum residue limits are proposed for approval. So unfortunately, the maximum residue limits for most recommended pesticides on various crops are not still available. So as fact of the matter, the maximum residue limits vary as per the recommendations in good agricultural practices as well. And the recent Joint Parliamentary Committee formed under the chairmanship of looked into the details of the tolerance for pesticide residues in bottled water and other beverages after the news report on pesticide contamination in water and colas has become an issue. So the set maximum residue limits for food, including the water, has also been done. Coming to the principles and methods of pesticide residue analysis. So we know pesticide residues are present everywhere, and their mechanisms we have seen, the management we have studied, and how what are the different departments that are working on maintaining these standards in foods and water has been just said. So so we know like we have certain platforms to deal with like JMPR and also how do you estimate the pesticide? Okay, I'll give you, for example, I'll give you grapes and I'll give you pomegranate and I'll give you apple or some other vegetables. And I'll ask you like which is having the highest pesticide content in it, like highest concentration of the pesticides in it. So how do you tell me that? By just looking by just looking at the fruit you can't, or by just looking at the superficially to the vegetable and the fruit you can't see, yeah, this much concentration is present, this much is concentration is present, so you can't see like that, but you have to use different methods to get to know about the concentration. So you have to use certain qualitative and quantitative methods to know the pesticide residues and to analyze them. And the objective of pesticide residue analysis is to study the kinetics of pesticide disappearance and to study the pesticide dissipation pattern in or on the plant, in or on the plant. So you have to do sampling, sample preparation, extraction, cleanup, identification and quantification, analysis of the control situation samples, fortification and recovery trials, confirmatory analysis, data interpretation. So these are the identification and determination of pesticides has to be done using this all parameters. Then sample analysis qualitatively and quantitatively has to be done. So after the sample processing has been done using by following all these steps, then that sample has to be analyzed for identification and quantification of various groups of pesticides. So there are various methods available for the identification of the pesticides. There are spectrophotometry, chromatography, enzyme-linked immunosorbent assay ELISA, and biological methods. And coming to spectrophotometry, it is useful for quick, relatively simple analysis of pesticides that absorb light in the UV visible range, both qualitatively and quantitatively. And chromatography, it uses gas chromatography and HPLC mostly is more suitable for complex mixtures of pesticides, offering high precision and sensitivity for both qualitative identification and quantitative measurement. GCMS, HPLC-MS or LCMS uh, enhances the accuracy and sensitivity of the pesticides. Coming to the principles of chromatography, the type of chromatography is adsorption chromatography, column, gas liquid, partition, and ion exchange. So in adsorption, you'll separate based on the attachment onto the solid stationary phase of that pesticide onto the solid surface, that is stationary phase. In column, sample components are separated as they pass through a packed column. It is like a silica gel or a C8 or C18 column. So they are like different columns used for different kinds of pesticides to be eluted. Gas liquid chromatography separation is based on the different interactions with the liquid stationary phase and a gas mobile phase. Okay, in partition chromatography, separation is based on the partition between two immeasurable phases, liquid and liquid. Here in partition chromatography, you will have the stationary phase liquid and the mobile phase. So in every chromatography, you will see a stationary phase and a mobile phase, whether it can be a solid, liquid, or a gaseous one. Ion exchange chromatography, the separation is based on the ion exchange interaction with the charged stationary phase. So each type of chromatography has specific use depending on the nature of the sample and the components being separated. The gas liquid chromatography is particularly used for volatile compounds, while ion exchange chromatography is widely used for separating charged species like ions and proteins. And partition chromatography and adsorption chromatography are commonly used for both liquid and solid sample analysis. So there is like a normal dissipation, like where your pesticide is seen, the identification detection is done, and if you use GC or HPLC, then you get a chromatogram where you can see here the chromatography methodology. So that is a chromatogram, how you will get the peaks, and by studying these peaks, then you will know the concentration. By calculating these peaks, then you can say the concentration. Whereas in the spectroscopy methodology, you will get the picture like this. So coming to the column chromatography, so this is how the column chromatography looks, and on the side I have given the real image in the lab that they usually do, where they put the sample, the load the sample, stationary phase is there in the column chromatography, and you tend to separate the compounds here. So it consists of a glass column of 1 to 5 centimeters in diameter, and gas column should be packed tightly with a powder-sized dry particle, that is the inert phase. If a thin layer of a sample mixture of two compounds is added to the top of the inert phase, and a liquid which is more strongly absorbed to the inert phase than any of the compound is the sample. So this process is called elution, and as the compounds move down the column, the small differences in their shape, charge, distribution, and polarity will allow them to be separated one after the other, and you have to collect them into different test tubes as shown here. Coming to thin layer chromatography, it's a chromatographic separation done on a thin layer of an adsorbent is called thin layer chromatography. It can be a paper that can be used where you put a dot of the samples and then you leave it in the solvent, and then depending on the rate of the flow and the this is followed by the visualization. So based on the absorbed chemicals on the solid stationary phase are pulled by the mobile liquid phase, and difference in this in the adsorption capacities or coefficient cause the separation. So in this technique, cleaned up extract is spotted along with a standard pesticide on a thin layer of absorbent material like a silica gel or aluminum coated on a glass plate, and the separation is achieved by ascending development by mixture of the solvents. So this is followed by visualization either by spraying chromogenic reagent or exposure to UV light. So side you can also see the image from the lab. Coming to the gas chromatography, it's a partition chromatography in which the mobile phase is gas. The sample should bear the following characteristics in order to be analyzed in a gas chromatography. Like it should be volatile, means that GC can separate any compound that boils without decomposition below 350 degrees only. And this represents about 15% of all the known components or compounds. And it should be thermally stable and should be detector sensitive. But in HPLC, this is not the case because non-volatile and thermally unstable compounds can be detected in HPLC. That's the main difference between a gas chromatography and HPLC. So this is the structure where you have a gas chromatography with glass flow regulator, carrier gas, sample, injector, column, and the column will change depending upon the pesticide that you are using, and detectors will also change based on the pesticides, and then the analysis will be done. So there are different types of GC detectors like FID, TCD, MSD, ECD, NPD, and FPD. We will be dealing with them now. So coming to the GC FID, GC FID means FID is a flame ionizing detector. So the organic compounds burning in the flame produce ions and electrons which can conduct electricity through the flame. So that is the basic principle here. Because of its relatively high sensitivity to most organic compounds, the flame ionizing detector is very powerful tool for the GC. So flame ionizing detector is the best for compounds containing carbon-carbon or carbon-hydrogen bonds. Example, fatty acids, flavor components, carbohydrates, and antioxidants. And coming to thermal conductivity detector, it's also called as nitrogen phosphorus detector. And these detectors are more responsive to phosphorus than to the nitrogen. And these detectors mainly depend on the hydrogen flow and the magnitude of the current supplied. The advantage is it's non-destructive to the sample and allows further analysis, but it's not that sensitive. Electron capture detector is highly sensitive detector capable of detecting the picograms amounts of specific types of compounds, and high selectivity of this detector can be great advantage in certain applications. So based on the application also, you will decide it. It's highly sensitive to molecules containing electronegative functional groups such as halogens, peroxides, quinones, and nitro groups, and ideal for pesticide residues. It is non-destructive, however, detection range is small. Coming to flame photometric detector, the reason to use more than one kind of detector for gas chromatography is to achieve selective or highly selective detection. And this device uses the chemiluminescent reactions of these compounds in a hydrogen air flame as the source of analytical information that is relatively specific for substances containing these two kinds of atoms. So basically, this increases the sensitivity of the detector can be increased by increasing the voltage, but an upper limit is imposed by the simultaneously increasing the noise. And the optimum voltage can be determined by comparing detector response to an amount of compound. So these are all the GC detectors that we have been seeing. And coming to the high performance liquid chromatography. So initially, while we are talking, we have said like GC is for the compounds. So one major difference that you have to remember is GC is for the compounds that are volatile, thermally stable, but in HPLC, non-volatile and thermally unstable compounds are separated. Like vitamin D3, you can vitamins, they are not thermally stable, right? So they're sensitive and they're non-volatile. So that can also be uh, done using the high performance liquid chromatography. It's a form of liquid chromatography in which effective separations are achieved through improvements in the technology. The major parts are the solvent reservoir, injector, analytical column, detector, recorder, and mobile phase. So mobile phase carries the sample through the column and helps separate the components based on their interaction with the stationary phase. Pumping system delivers the mobile phase under high pressure and at a constant flow rate to ensure smooth operation to the system. And columns, so different columns are used even in this, like C8, C18. So different kinds of columns are used based on the compound that you're going to elute. Okay. And detection systems, it detects the quantifies the separated components as they elute from the column. And the mobile phase also differs. The percentage of the mobile phase also differs. The solvents also differs based on the compounds or the pesticides or any other vitamins. Based on that, you have to change the mobile phase. You have to change the column. You have to change the flow rate. You have to check the uh, uh, what you call where the peak you are getting, uh, where because the retention time is also important for this, right? So the retention time of each compound is different. If you'll get vitamin D at 8.3, then it might be different for different vitamins. So you have to standardize all this methodology. You have to standardize the flow, mobile phase. You have to standardize the rate of the flow, and you have to standardize many things in this. So it's not as easy as you see, but yes, these are all like very sensitive techniques. And other than HPLC, you also have LCMS, the uh, higher and sensitivity that it is there for HPLC. Whereas for GC, you also have GCMS, the mass spectroscopy method, where it is like more sensitive and more uh, reliable as well. So with this, you will be concluding the chapter 16 as well. And in this, we will see. I'll stop sharing it so you can see me. So in this, you will have model questions like what are the various objectives of pesticide residue analysis, and describe the principle of the chromatography, and describe the principle and use of commonly used detectors in gas chromatography. So we have seen in detail different detectors that are used in gas chromatography and their use, and what are the different types of compounds that are used for each detector. So that you have seen in the class, so that you have to write. So with this, we have finished the syllabus of the toxicology of insecticides, in which you have 16 chapters, 16 units. So initially we have dealt with the block one, that is the fundamentals of insecticide toxicology in your syllabus. In that you have seen the formulation and classification of insecticides and evaluation of insecticide toxicity, insecticides and cuticle, and effects of insecticides on the nervous system. And coming to block two, the botanicals and conventional insecticides, we have dealt or we have known about the botanical insecticides, especially nicotinoids, pyrethrum, neem, rotanin, and retinoids. If you have not, if you have missed any of the classes, please go to the YouTube where you have live classes uploaded there. So you kindly go through that and check out the videos for better understanding, and then look at the syllabus, and also I've mentioned model examination questions at the end of every unit, so kindly go through it. So in block 2, we have dealt with botanical insecticides, organochlorines, organophosphorus insecticides. I'm just reading out your syllabus, what all we have covered, and carbamate insecticides as well. In block 3, we have seen the newer insecticides like synthetic pyrethroids, molecules with different chemistry like neonicotinoids, and synthetic insect control agents. And coming to block 4, we have metabolism, resistance, and residues. And in that, we have studied the photochemistry of the pesticides, the basic principles in photochemistry, factors affecting the pesticides. And we have seen in yesterday's class, we have seen the microsomal monooxygenases and the role of CYP450s. And we have also dealt with the extra microsomal metabolism of the insecticides. And in today's class, we have seen the insecticide resistance and how the development of the insecticide resistance happens, the mechanisms of the insecticide resistance, the factors conducive to rapid development of the resistance, and genetic basis of resistance, and management of the insecticide resistance. And now we finished the pesticide residues. Basically, the critical terms and definitions: biomagnification, bioconcentration, and bioaccumulation, effects of pesticide residue on human beings, environment, and non-target animals, and pesticide residues and standards in foods and water, and principles and methods of pesticide residues. How do we analyze them using qualitative and quantitative analysis? How do we prepare the sample? And what are the different chromatography and spectrophotometry techniques that we use? And what are the different kinds of GC, gas chromatography, and HPLC chromatography methods that we use? And what are the different kinds of detectors that we use for different types of compounds has been studied along with the HPLC. So coming to the model question paper, so I just want to roughly give you the idea of it. So you have that in your book. So initially in a section A, you have 4 marks questions. 4 questions you have to write, and that has 5 marks. So basically, what are the different criteria for classifying the insecticides? What are the different neurotransmitters found in the insect's nervous system? What do you understand by the type 1 and type 2 pyrethroids? Write an essay on the use of insect attractants. So these are all the short questions, and out of 8 questions, you have to answer any 4. And coming to the section B, you have to answer any 5 of the questions out of 10 questions in about 30 lines each. Okay, so you have 10 marks for each question. So if you answer 10 questions, you will be getting 50 marks. So you'll have choice of 5 questions out of it. And section A, you will have 8 questions that are short answer questions, which has to be written in 10 lines. And out of 8 questions, you have to answer 4 questions. So 4 into 5 equal to 20 marks. So total, your marks will be for maximum marks is 70. Okay, so I guess I've covered it. So in the 30 line questions, give a detailed account of probit analysis. Yeah, that we have dealt. So you can go into our previous classes. So you just go through all the classes. Finish your syllabus. So we are done with the syllabus. We are given like 10 classes for this course. That is course 7 for the toxicity of insecticides. So in 10 classes, we are able to finish 16 units. So all the 16 units classes are uploaded in the YouTube. Kindly go through it. And for better understanding, I have given the references of each and every image that I have adopted from the recent journals that I have gone through. So if you're interested and if you're interested in reading or getting to know, have more knowledge in that, so you can just go copy paste that DOI of the reference that I have provided. You can directly go and copy paste that in the Google, and you will get the full length paper of that particular authors of that particular journal. So you can go through that on get more detailed explanation for each and everything that we have covered in this syllabus. So with this, we are done with the toxicology of insecticides and its syllabus. Thank you.